So this video covers treatments for infertility. Okay. So we're going to look at the way that drugs can be used to stimulate ovulation first of all. Right. So lack of ovulation can cause infertility. And this is normally due to a failure of the pituitary gland to release sufficient FSH and LH to stimulate follicle development and therefore ovulation. And in this case, ovulation is stimulated by drugs to bring about ovulation and hopefully a successful pregnancy. So, the way ovulation stimulation drugs work is by preventing the negative feedback effect of estrogen on FSH secretion. So if you think back to the previous key area where we talked about the hormonal control of the menstrual cycle, you'll remember that high levels of estrogen have an inhibitory effect on the pituitary gland and prevent the release of FSH. This then prevents the development of any more follicles within the ovary. If you give a woman drugs that prevent the secretion of estrogen, that means that levels of FSH produced by the pituitary will remain high, and that will stimulate the development of multiple follicles within the ovary. You can also give drugs to mimic the action of FSH and LH and bring about ovulation that way. These drugs will stimulate the development of several follicles during one menstrual cycle, and this can result in something called superovulation. That is, many eggs being released from the ovaries at one time. Okay, so if you get multiple eggs released from the ovary, and they are all fertilised by a separate sperm, that can then result in multiple births. Now, this is very useful if this is done before in vitro fertilisation, because that can enable multiple eggs to be collected all at once. If it's being done as part of different fertility treatment, it can result in multiple births, twins or triplets or even more. Okay. So now I would like you to pause the video and answer the questions on ovulation stimulation. So ovulation stimulation is used when there is failure to ovulate or due to hormone imbalances. Ovulation is stimulated using drugs which inhibit estrogen, which will then increase the release of FSH by the pituitary. Effects of ovulation stimulation are multiple follicles will mature. Side effects are superovulation, which can result in multiple pregnancies. Okay, next we're moving on to talk about artificial insemination. So you need to be able to describe the process of artificial insemination and explain when it is used. So, artificial insemination is the introduction of semen into the female reproductive tract, usually so this next part so this section covers artificial insemination. And you need to be able to describe the process of artificial insemination and explain for what conditions it is particularly useful. Insemination is the introduction of semen into the female reproductive tract. It occurs naturally during sexual intercourse. However, it can also be carried out artificially. And artificial insemination is when the semen is inserted into the female reproductive tract by some other means than sexual intercourse. And you can see in the diagram here, they've put a thin tube called a catheter up through the vagina, through the cervix, into the uterus, and that is being used to deliver the semen into the uterus. Okay, this is a much clearer diagram, right, showing the vagina being held open by a speculum. You've got a syringe containing semen samples, and the catheter has been inserted through the cervix into the uterus, and that is being used to deposit the semen into the uterus. Artificial insemination is used where the male has got a low sperm count. And what they'll do in that case is they will take several semen samples collected over a period of time, and these will be frozen until they are required. Then those samples are defrosted and inserted into the uterus using the method we saw in the previous slide. Right. If the male partner is completely sterile, then a sperm donor may be used to provide the semen. Okay, so pause the video now and answer the questions on artificial insemination. So, artificial insemination is used where the male has a low sperm count or other problems with sperm. The semen is collected over a period of time and several samples are usually collected. If a partner is sterile, a donor may be used. And here are some video clips you can watch describing artificial insemination if you would like some more information. Right. Fertility treatments can sometimes result in multiple births, right? Particularly if ovulation drugs are used, quintuplets, 5 little babies there. Right. She had intrauterine insemination or artificial insemination. And this resulted in quintuplets because she took drugs to stimulate multiple ovulation. There we go. Okay, next we have something called intracytoplasmic sperm injection, which is abbreviated, thank goodness, to ICSI, I-C-S-I. Okay, and again, you need to be able to give a description of the process of ICSI, explain what happens, and explain when it is used. So, ICSI involves injecting the head of a sperm directly into the egg to achieve fertilisation. It needs to be done under a microscope so that the embryologist can see what they're doing. Okay. So you can see here a very fine needle is used. There is a pipette to hold the egg in place. The needle is inserted through the membrane of the egg into the nucleus, and the head of the sperm is injected directly into the nucleus of the egg. Okay. This is done using a powerful microscope, 8,000 times magnification. The embryologist has to select healthy sperm, so they're not looking for sperm with abnormalities. You can see that these ones here have got misshapen heads. This one here, the nucleus is a bit too small, but this one here has got a normal shaped head and nucleus, so this one is going to be chosen to be used in the process. So ICSI would be used when there is either a high proportion of defective sperm, so lots of sperm have got abnormalities, or where the sperm count is very low. Okay. It can also be used when genetic testing or pre-implantation genetic diagnosis is being performed, and we're coming to that in a minute. And it's also a good option for people who have problems with fertilisation, and it's very commonly used now as part of IVF, which again we're going to come to in a moment. Okay. So once the egg has been injected with sperm, the egg will be left with nutrients and it will be allowed to divide. It will be observed by the embryologist as it divides, and if the embryo that's produced is of a good quality, it will then be transferred into the female's uterus. Right, so now's the time to pause the video and answer the questions. ICSI stands for intracytoplasmic sperm injection. It is used during IVF. It's used if the sperm are defective, or it's used if the sperm are very low in number. And the technique is when the head of a sperm is drawn into the needle and it's directly injected into the egg. Okay, this brings us on to IVF, or in vitro fertilisation. Okay, so you need to be able to explain what IVF is, and explain that it's often used in conjunction with pre-implantation genetic diagnosis. So IVF happens when fertilisation takes place outside of the female's body. Okay, eggs are generated by hormone stimulation. They are removed from the female's body. Fertilisation takes place in the lab, and then any healthy embryos are implanted back into the uterus. It comes from the Latin in vitro, means in glass. Okay, so the first stage of IVF is egg retrieval. Okay, the woman will be given fertility drugs to stimulate multiple follicles to develop with multiple eggs inside, and then under ultrasound, a probe will be used to remove the eggs from the ovary. So they use a needle, and they look at the ultrasound picture and they detect where the mature eggs are, and they use the needle to withdraw the mature eggs from the follicle within the ovary. Okay, so like I said, they use hormone therapy to stimulate the development of lots of follicles within the ovary, and then they collect those eggs. They then fertilise the eggs in a culture dish, or quite often nowadays by ICSI, to create several embryos. Okay, those embryos are incubated until they've got at least 8 cells, and if the embryologist considers them to be healthy enough, they will be transferred into the uterus for implantation. Okay. Again, this is done via ultrasound to guide the doctor. Okay, and they use a thin, flexible tube called a catheter, which goes up through the vagina, through the cervix and into the uterus, and a syringe is used to deliver the embryos into the uterus. Okay, so 1 or 2 embryos are transferred to the uterus, and hopefully implantation will take place and a pregnancy will occur. Okay, however, it's not guaranteed that pregnancy will be occurring. So, if there are more embryos produced than are needed, then they can be frozen and they could be used again for subsequent attempts if necessary. So, to summarise what takes place in IVF, the natural menstrual cycle is suppressed with medication, and then drugs are taken to stimulate ovulation. That then means that several eggs develop at once. Those eggs are then collected via a surgical procedure whereby a needle is used to withdraw them from the ovaries. The eggs are then fertilised, and then they are allowed to divide to produce embryos of at least 8 cells, and then the embryos are transferred to the uterus via a catheter. And this diagram summarises the process.